Okay, so what I'm going to do is go over the rest of protein synthesis, which we haven't done in class, and then I'll go over chromosomes, and then what we can do is we can start on gene control and mutations next time. Alright, so we remember that we have 20 amino acids which make up, combinations of these make up our proteins. We have things called codons, which are a triplet or 3-base pair, or three bases that code for different amino acids, and here's the little table there. So I'll just get rid of myself real quick, and here's our final amino acid, glycine. But yeah, that's our little table. So what we'll do is we'll start off with a little brainstorm. So first question is, why is it essential to specify so accurately the primary structure of a protein? Also, what is a codon? What chain of mRNA bases would complement the following DNA sequence? So what's complementary to A, C, 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 T, C? And this question is really to get you thinking. So if you remove the nucleus from a single-celled organism, it continues to synthesize proteins for at least 2 weeks. How is this possible? So pause this now and have a go at answering these questions. And here are the answers. So the primary structure of the protein determines its shape, which in turn determines its function. Is what you should have got there. A codon is a triplet code of bases, which codes for a particular amino acid. Then we have the UGG GAG is complement to what we had on the previous slide, which was ACC CTC. And the final question was, if you remove the nucleus, why are proteins still synthesized? Because there's still mRNA in the cytoplasm, and this can be translated many times. So remember, DNA is transcribed into mRNA, and that bit of mRNA can be translated into proteins again and again and again and again. So even though the nucleus is removed, protein synthesis can still occur. Okay, so we know that we have DNA, which is transcribed into mRNA, then that is spliced in particular ways to create mature mRNA. Then that mature mRNA is then translated, so it's read through ribosomes, and those ribosomes sort of is where the polypeptide chain of the proteins is built. And you've got mRNA, then you've got transfer RNA, which goes out and gets the amino acid. So that's what we have here. So we've got our ribosome, and we have our mRNA strand, and we have our corresponding tRNAs. And so the ribosome processes the mRNA, and the transfer RNAs have the anticodon to the mRNA, and then that anticodon is specific to a particular amino acid. So the transfer RNA goes out and gets all the specific amino acids, and they match up to the complementary codon and anticodon system, and the ribosome joins those amino acids together and starts to build the polypeptide chain. Then the ribosome moves along the messenger RNA. The transfer RNAs are then released, but the amino acids stay joined together and begin to form the polypeptide chain. So that's what's going on there. So this slide here is translation in a nutshell. So again, you have the mRNA along the bottom, that is threaded through the ribosome. Then you have the transfer RNAs going out and getting the specific amino acids. The transfer RNAs have an anticodon, which is complementary to the codon found on the messenger RNA, and that determines the order in which the amino acids are linked in the polypeptide chain. That polypeptide chain then begins to fold, and as the entire length of the mRNA is read off, we have our protein. So hopefully this is all familiar to you. So a quick recap. So DNA is the double helix. It keeps the genetic code neatly in the nucleus. It's nice and safe there, so there's no permanent changes in the DNA. The nucleotide is the deoxyribose plus the phosphate group. So the deoxyribose is a sugar, the phosphate is a phosphate, and that creates the phosphate sugar backbone, and then we have our nitrogenous base. And the bases are adenine, thymine, guanine, and cytosine. And the complementary base pairing allows DNA replication and mRNA transcription. Right, and now I'll recap on RNA. So you have RNA is single-stranded, unlike the DNA, which is the double helix. So the nucleotide equals the ribose, which is the sugar, which is different to deoxyribose. Ribose has an extra sugar, plus the phosphate, and again that creates the phosphate sugar backbone, plus the nitrogenous base. And the bases are adenine, uracil, guanine, and cytosine. Messenger RNA takes the genetic code to the cytoplasm. You've got rRNA, which is a part of the ribosome, presents the codon for reading, and then you have tRNA, which places correct amino acid in the sequence. So proteins and polypeptides. Amino acids are the monomers, the building blocks for the single units. We have 20 different amino acids that are commonly used. The structure of the polypeptide and thus the protein is very important to its function. And one gene codes for one polypeptide. Alright, so here are some keywords that are important. So transcription, translation, mRNA, tRNA. rRNA is just ribosomal RNA, RNA found in the ribosome. Don't worry about that too much. Codon, anticodon, RNA polymerase, introns, extrons. Remember you've got junk DNA, splicing, triplet, stop and start codons, which we haven't gone over in class. But that's fine. So answer these questions from the quick quiz. So just pause this now, I'll give you a minute to do that. I'll give you the answers. Okay, so here are the answers to the quick quiz. Fingers crossed you got all those right. And now we're going to move on to chromosomes.